Carcinogenesis refers to the process through which normal cells transform into cancerous cells. It is a multi-step process that involves genetic mutations, changes in cellular signaling, and alterations in the environment surrounding cells, ultimately leading to uncontrolled cell growth and the formation of tumors. Carcinogenesis is a complex biological process that can span many years and may involve various stages, from initiation to progression. Stages of carcinogenesis. Carcinogenesis is often described in several stages. 1. Initiation. This is the first stage where a normal cell undergoes a genetic mutation, typically caused by exposure to carcinogens. These mutations may affect genes that regulate cell growth, such as proto-oncogenes or tumor suppressor genes. 2. Promotion. After initiation, the mutated cells may undergo clonal expansion, a process where the mutated cells proliferate. The promotion stage is often driven by factors that encourage the growth of initiated cells. These factors may include environmental influences such as hormones, chemicals, or viruses. 3. Progression. In this stage, the initiated cells undergo further genetic changes that increase their ability to divide uncontrollably, evade cell death, and invade surrounding tissues. This stage often results in the formation of malignant tumors with increased invasiveness and the potential to spread, metastasize, to other parts of the body. Factors responsible for carcinogenesis. Carcinogenesis is influenced by both external, exogenous, and internal, endogenous factors. 1. Environmental factors, exogenous. These are external agents that can cause damage to cellular DNA and lead to mutations. Chemical carcinogens, substances that can cause cancer through direct or indirect DNA damage. These include tobacco smoke, contains carcinogenic chemicals like benzene, formaldehyde, and polycyclic aromatic hydrocarbons. Asbestos, a fibrous material that can cause lung cancer and mesothelioma when inhaled. Aflatoxins, produced by molds in improperly stored grains and nuts. These are potent carcinogens linked to liver cancer. Alkylating agents, chemicals used in chemotherapy, which can cause mutations in DNA. Radiation, exposure to ionizing radiation, e.g., X-rays, gamma rays, can damage DNA by causing breaks in the DNA strands, leading to mutations. Non-ionizing radiation, like ultraviolet UV light, can also damage skin cells and lead to skin cancer, e.g., melanoma, basal cell carcinoma. Physical agents. Certain types of physical trauma or chronic irritation can contribute to carcinogenesis, though they are less direct than chemical or radiational factors. 2. Infectious agents. Some viruses, bacteria, and parasites are known to contribute to carcinogenesis. These agents may introduce their own genetic material into the host cells or cause chronic inflammation, leading to mutations. Viruses. Certain viruses are implicated in the development of cancers. Examples include human papillomavirus, HPV, a major cause of cervical cancer and other genital cancers. Hepatitis B and C viruses, chronic infections lead to liver cancer. Epstein-Barr virus, EBV, associated with several cancers, including Burkitt lymphoma and nasopharyngeal carcinoma. Human T lymphotropic virus type 1, HTLV1, associated with adult T-cell leukemia. Bacteria, Helicobacter pylori, a bacterium that causes chronic gastric ulcers, is linked to gastric cancer. Genetic factors, endogenous. Inherited genetic mutations can predispose individuals to cancer, and certain genetic alterations can be passed down through generations, increasing the risk of carcinogenesis. Oncogenes. These are mutated or overexpressed versions of normal genes, proto-oncogenes, that normally control cell growth. When activated, they can drive uncontrolled cell proliferation. Tumor suppressor genes. These genes normally act to inhibit cell division or promote apoptosis, programmed cell death. Mutations in tumor suppressor genes, like the p53 gene, can prevent the cell from undergoing apoptosis, allowing abnormal cells to survive and proliferate. DNA repair genes. Defective DNA repair mechanisms can result in the accumulation of mutations. For example, mutations in the BRCA1 or BRCA2 genes significantly increase the risk of breast and ovarian cancer. 4. Lifestyle and dietary factors. Dietary habits, exercise, and other lifestyle factors can influence cancer risk by affecting cellular processes and gene expression. Obesity. Being overweight or obese increases the risk of several cancers, including breast, colon, and endometrial cancers. Obesity is thought to alter hormone levels and create an inflammatory environment conducive to cancer development. Dietary factors. A diet high in red or processed meats, low in fruits and vegetables, and rich in fats may increase cancer risk. Conversely, a diet rich in antioxidants, e.g., from fruits and vegetables, can help protect against cellular damage. Alcohol consumption. Chronic alcohol use is a known risk factor for cancers, particularly those of the liver, mouth, throat, and esophagus. Physical inactivity. A sedentary lifestyle is linked to an increased risk of developing several types of cancer, including colorectal and breast cancer. HA5. Hormonal factors. Hormones, particularly sex hormones, can play a significant role in the development of certain cancers, especially reproductive cancers. Estrogen. High levels of estrogen or prolonged exposure to estrogen, e.g., early menstruation, late menopause, or hormone replacement therapy, are associated with an increased risk of breast, endometrial, and ovarian cancers. Testosterone. Elevated levels of testosterone may be linked to prostate cancer. 6. Immune system factors. A weakened immune system can increase susceptibility to cancer, as the body's ability to detect and eliminate cancerous cells is impaired. Immunosuppression. Individuals who have weakened immune systems due to conditions like HIV/AIDS, organ transplantation, or autoimmune diseases are at higher risk of developing certain cancers, as their bodies cannot effectively recognize and destroy abnormal cells. Conclusion. Carcinogenesis is a complex, multifactorial process influenced by genetic, environmental, and lifestyle factors. Understanding these factors helps in the prevention and early detection of cancers. While some factors like genetics cannot be controlled, many lifestyle choices, such as avoiding tobacco use, maintaining a healthy diet, and protecting against radiation, can reduce the risk of cancer.